everybody. Thank you so much for joining. We are delighted to see so many people here on this about to be rainy day. I'm Maya Magnus from the Clinical and Population Sciences core of the DC CFAR, and I'm here with Larry, if you want to say hi.
Hello, everyone. I am the core manager for the CPS DC CFAR core.
She is the one who brings us all together so beautifully and helps manage this whole process. And of course, Dr. Sam Simmons. Sam, if you want to say hi.
Hi, good to see everyone.
And our fearless leader, Sedley, is saving lives on service today. So she sends her regrets. She might be able to pop in later, but she can't join us right now in this very exciting time. So I am not going to spend too long introducing our wonderful speakers, Dr. Kate Michelle and Dr. Mimi Ghosh. I can honestly say I love this topic. It was Mimi who really taught me the meaning of translational, learning about women and different factors that make them more susceptible to HIV and remarkable immunologic approaches in the cervicovaginal tract. And she really brought the light to me personally about how these people and basic science connect. And I think that's one of the hallmarks of the CFAR and one of the wonderful things. And I know Kate has done a lot of similar work trying to understand and untangle some of the risk factors for women. And Kate's from Georgetown and Mimi is from GW. And without further ado, I think we're going to have a talk from each of them of a good size, but then hopefully have enough time for questions. And first we'll go with Kate, and then we'll go with Mimi, and then we'll have some questions. I will, Larry, Kate, is she shareable?
Yes, she can share her screen.
Oh, great, because I don't think I have the host. Okay, great, wonderful. Well, thank you so much for joining us, and take it away, Dr. Michelle.
Great. Thank you all so much for the invitation to talk to you all today. So I'm going to be speaking more broadly about the cervicovaginal microbiome in the context of HIV, and I'll just squeeze a little bit of research in on cervical dysplasia as well. So just really briefly, first, what is the cervicovaginal microbiome? So first, when we talk about cervicovaginal, we're really talking about the ectocervix and the vagina. A lot of the time, mucus plug kind of prevents a lot of access to uterus, although uterus is by far not exclusively sterile by any means, just kind of a different microbiome that we're going to be talking about. And microbiome, what very, very briefly, high level, it's just the collection of microbes that live within the cervicovaginal environment. This includes bacteria, yeast, fungi, including yeast, other eukaryotes, but predominantly it's bacteria, and that's what most of the research focuses on and what I'll focus on today. So what types of microbes live in the cervix and vagina? I'm going to talk about this paper from 2010, which is really the first work that really took a molecular approach, meaning they sequenced all of the DNA of bacteria within the cervicovaginal environment. And it was a large study of nearly 400 girls and women, and it was racially and ethnically diverse. So although not geographically diverse, mainly in Baltimore and Atlanta, but I want to note here too, I think I mentioned it a second ago, but this was in 2010. So this is still a field, microbiome research itself is still pretty much in its infancy, but the vaginal microbiome is even more, we haven't really fully delved into all of the dynamics. So within the study, I want to present this figure, but just very quickly so that we don't kind of get overwhelmed with this, I just want to talk very briefly about how to interpret this. Because a lot of microbiome papers, you'll see these kind of heat maps, and I think they can be a little intimidating. So across, bars across are just talking about one bacteria type. So this across the top is L. vaginalis, and then we have different bacteria types as rows here. In the columns, there's very thin lines that represent a sample from one person, and then they're color-coded based on the bacteria abundance. So red here would be more bacteria, and yellow would be less of the bacteria. And then there's kind of this color scale in between. Okay, so back to this figure. They kind of noticed that there were distinct clusters, and they actually looked at this statistically. There were clusters of the bacterial communities in the vaginal microbiome, And about 73% of the samples were dominated by a bacteria called Lactobacillus. Different species, Lactobacillus is the genus, but Lactobacillus itself was the main component for most. And as you can see here with, again, the red indicating high abundance, two of the major groups were Lactobacillus crispatus dominated or Lactobacillus iners dominated. And now if we expand this and kind of look at just a little bit further down on the same heat map, about 27% of the samples were in what, for lack of a better term, just kind of called this high diversity group. There wasn't really one bacteria that seemed to be far and away the main component. You kind of see these kind of purplish pinks of just kind of a spread of bacteria that tended to be more anaerobic. And while these samples still had lactobacillus at lower levels in most cases, they weren't the dominant bacteria. So what else did we learn from this paper? Or what do we care about in this high diversity group as well? So they also found that dominant bacteria associated with vaginal pH. And this is because lactobacillus, so named because it produces lactic acid as one of its main metabolites. So we can see from this table from the same figure, across all ethnic groups, those with Lactobacillus crispatus dominance had a very low vaginal pH. Those with Lactobacillus iners dominance had kind of an intermediate pH, and those in the high diversity group had a higher pH. And this is important because generally pH is associated with a higher pH with more pathogen persistence. A lower pH kind of eliminates some of these pathogens that's thought to kind of act as almost a natural barrier against pathogen invasion. They also found that the high diversity group associated with clinical diagnosis of bacterial vaginosis. In this paper, I think they used Nugent scoring, but essentially a clustering of symptoms that, kind of associated with malodor, pain, etc. And this kind of gets at this kind of difference, I think that's still being hashed out in the literature between kind of a clinical diagnosis of BV, which is more based on symptoms, versus molecular BV, meaning that we kind of find this high diversity group at the DNA level, regardless of what your symptoms are actually maybe presenting, or maybe you don't have any symptoms at all. But it was interesting because this was one of the first papers that kind of established this. Many papers since then have kind of found all of these same associations relatively held up. So how does the cervicovaginal microbiome affect our health? Well, first off, vaginal dysbiosis. There were a cluster of papers in the early to late 2000s that kind of found that a clinical diagnosis of BV, mostly, I think most of these papers were using Nugent scoring, associated with HIV acquisition. And the interesting thing in this study from 2005 is that a normal flora, meaning more lactobacillus dominant, was kind of the reference here. But if you had an intermediate flora, just kind of in between normal and this high diversity group, you got a bit of an increased risk for HIV acquisition, which increased even further in those with a clinical diagnosis of BV. And this has been found in other papers as well. And additionally, in some papers with molecular diagnosis. So there have been some people finding this association with vaginal dysbiosis associating with HIV acquisition, and further BV or molecular BV associating with a higher cervicovaginal HIV part load. And one paper in 2009 found that they found that 29% of incident HIV infections were attributable to BV in a large cohort of women. But BV itself is associated with a lot of adverse health outcomes that I'm not going to get into too much detail here, but associating with other STIs, gonorrhea, chlamydia, PID, preterm birth. An interesting paper from just a couple of years ago from Nikki Platt that kind of did a reanalysis of the CAPRISA 004, found that the vaginal microbiome can affect PrEP efficacy as well, which was pretty surprising. So they did this reanalysis, and this was a topical vaginal tenofovir gel, and they found that it reduced HIV incidence. I think if you all remember, there were kind of overall lower than expected rates from the trial, and so they broke it out by looking for molecular BV. So 61% had reduced HIV incidence under a lactobacillus dominant vaginal microbiome, but the effectiveness was only 18% for those with a non-lactobacillus dominance. Further, they actually tested mucosal levels of tenofovir, and they were found to be lower in non-lactobacillus dominant women. And a graph shown from this paper as well, showing that they actually took these bugs and cultured them separately with tenofovir and found that within about 4 hours here, Gardnerella vaginalis was able to essentially start reducing and breaking down tenofovir, while those with Lactobacillus anners and Lactobacillus crispatus retained, there wasn't really any breakdown of tenofovir in culture as well. So that was a pretty interesting study. There's a lot of dynamics. Lactobacillus is not completely complacent. There's another recent paper that showed that BV is able to metabolize metronidazole, which is a treatment for BV. So the bacteria, more or less, bacteria are doing a lot more than just kind of modulating vaginal pH. They're also there potentially interacting with any drugs that might be present as well. So they have a really fascinating role in cervical vaginal health. But just to kind of focus back on, this interesting association with higher cervical HIV viral load. And this is kind of my segue into talking a little bit about some of the work that we've done within the WISE cohort. And we have formerly WISE, now MWCCS, still have to get used to that. But some preliminary data we found that women with HIV who naturally control, particularly CD4 cells, and as well among those who control HIV viral levels without ARVs, tend to have more cervical vaginal lactobacillus. And we haven't really been able to figure out any kind of mechanism for this, and we're still working on, a little delayed with COVID, but further studies to try to understand some of the relationships that may underlie this. But essentially, lactobacillus dominans was found in almost all CD4 controllers, those that maintained a high CD4 count without ARVs. And 45% of viral controllers, meaning they controlled HIV virus without ARVs, compared not only to women with HIV taking medication, but also women without HIV. And kind of highlighted here in green, the lactobacillus groups here. And I also want to highlight I should have mentioned earlier, Gardnerella vaginalis is sometimes implicated as one of these high diversity groups. It's sometimes a kind of linchpin. It creates a lot of proteins that are known to kind of interrupt the vaginal epithelium itself. One of the interesting things is that CD4 controllers, none of the samples we had, although it was only 8 total samples, none of them had Gardnerella vaginalis dominant or any high diversity sample really at all, which was quite surprising to us. If you take a cross section of any 8 or 10 women, chances are some of them may have a molecular BV diagnosis. I believe it's about 30% of women in the US, at least at any given time. So that was a little bit surprising to us. And when we kind of did a mixed modeling on this, lactobacillus dominant significantly was enriched among CD4 controllers. And I want to relate to, this is a PCOA plot. So it's essentially just, you can interpret this as points closer together are more associated, points further apart are not as associated. But kind of illustrated somewhat on the previous graph, but I wanted to just make it a finer point in this graph. We didn't really see any differences in the vaginal microbiome by HIV status as well, which was very interesting. We also have a separate study investigating how the cervical vaginal microbiome may affect cervical dysplasia progression. This is also a separate study of the WISE cohort, and we followed women with a cervical dysplasia diagnosis at CIN2, cervical intraepithelial neoplasia 2, kind of a diagnosis where for a lot of times treatment may or may not be prescribed. It's part of the reason that we picked that diagnosis. We followed them forward in time, and we measured at a few time points HPV types, vaginal pH, BV, and some other markers as well. Looking at the outcome of either CIN2 still remaining or progressing over time, or the CIN2 regressing to either CIN1 or normal. And here I just have a Kaplan-Meier analysis, but we interestingly found, and this is starting at CIN2 diagnosis moving forward, HPV type did not predict whether persistence progression or regression of disease would occur, which is kind of surprising. I definitely thought that, of course, HPV is going to be a driving force in driving progression or persistence of disease. But interestingly, we did find highly significant associations for BV, the presence of BV at CIN2 diagnosis, or vaginal pH at CIN2 diagnosis. We were a little surprised to find something this strong, but I think we're moving forward with trying to understand a little bit more about this, do some metagenomics, proteomics to try to figure out, what factors could be involved. But I think it's really interesting, and this has kind of been found. There have been a rash of papers in the last 5 years or so among women with not with HIV that have kind of found similar results. So we're moving forward with this as well. And I just wanted to end with a quick talk or quick discussion of how can we change the vaginal microbiome? If we think that it could be affecting health, are there any promising leads on how to potentially manipulate the vaginal microbiome? This is just a table from a very recent study. Although lactin B is a probiotic that's kind of been tested for a long time, I think it's moved into someone to be able to correct me, phase 2 or phase 3 at this point. But published results finding that bacterial vaginosis recurrence was a lot better or reduced among those who received lactin B, which is a probiotic topically applied to the vagina compared to a placebo, which is pretty exciting. BV is notoriously hard to treat. So look forward to more studies on this as well. Another more interesting areas, you may have heard of fecal matter transplants. There was a recent study looking at vaginal microbiome transplantation in the context of treating recurrent BV. And they found some preliminary results that were a little promising. I believe this is only 5 women, so it's not a massive sample size. And there are a lot of folks, curious about the ethical implications of these kind of fecal or vaginal microbiome transplants. But I think, potentially an area to watch and kind of see what happens in the future for this field. Okay, well, thank you all so much for letting me talk to a little bit to you about the cervical vaginal microbiome and a little about my research. I just want to thank my team, and thanks for the DCCFAR CPS Corps for letting me talk.
Thank you so much. We'll give you a virtual round of applause, Dr. Michelle. Thank you so much. I think we do have enough time for a few questions, and then we'll turn it over to Mimi, and then have questions for her, and then we can have questions for the both of them at the end. How does that sound? Are there any urgent questions people would like to raise right now? How about we go Jean, Rebecca, and then Deborah, and then we'll go to Mimi.
Thanks, Kate. I really enjoyed your talk. My question is around Trichomonas vaginalis, and I think some of your earlier data that you shared showed that there was not an association of an increased risk with HIV acquisition, and I always thought it was the opposite. So could you clarify that?
I think there was an increased risk with
Trichomonas, yeah.
Was it this one? Was it one of these tables that had trick in my?
That one.
Oh yeah.
That
okay.
slide.
Yeah, that's interesting.
Yeah.
Trichomonas at enrollment. It was longitudinal. I wonder if it was just looking at trick at enrollment versus longitudinally, maybe. Yeah, that is kind of.
Yeah, I just wanted to raise that point. Thanks.
As far as I know, I'm not an expert on trick really, but yeah, as far as I know, it should theoretically increased risk. Yeah.
Yeah, because of the inflammation.
Yeah. Yeah. Good point.
Great, Rebecca, did you have a question?
Hi, great talk. How did you define sin progression?
Yeah, so that's a great question. We looked at, and we have an OBGYN on our team that reviewed. I am more on the lab side than I am on the clinical side at all, who kind of reviewed based on, there must have been an inadequate colposcopy and adequate biopsy. Let me think there are other criteria. And then any progression, we didn't set, and I think this might have been a little bit of a limitation, but we didn't set a time, a time limit there. We have about 12 and a half years of follow-up. So you may argue, is that still associated with the same HPV types? That could be part of the reason that we didn't see that association. But we.
So was the event developing sin 3 or regressing?
Yeah, it was either persistence progression, so either another CIN2 diagnosis or a CIN3 diagnosis. We didn't have anyone progress to invasive cervical cancer versus CIN1 or normal. Or for regression, we also included folks, I believe, who had no subsequent CIN2 or CIN3, but they must have had 3 consecutive normal paths as well. Those were lumped into regression. And I didn't mention this, but we actually had most of the women regressing as well. It was a little over 60%. So very high rates of regression. It was also very quick as well. Most women, I think, regressed within a year or so. So that was part of the reason that we wanted to do a little bit more exploring and try to figure out any factors that may be involved. Yeah. I hope I
did you do the HPVs like of the lesions, or did you just do a swab?
It was from the cervical vaginal lavage.
Oh, that's right, from the Ys. And then were there multiple HPVs? I'm good. Like, were there multiples, or is it predominantly certain types?
There were multiple. I should have included.
I was just kind of curious on the diversity.
Yeah, there were multiple types detected. It was interesting because we had surprisingly low HPV 16, 18, which I think some other folks have kind of found as well. That's not too shocking, really. But a lot of, I think one of the only individual HPV types that was marginally significant was HPV 35, surprisingly. There were high levels of HPV 54.
Well, 35 makes sense.
Yeah, not entirely surprising.
That's it. I don't want to dominate.
Oh no, you're
Thank
fine.
you.
Yeah,
Thanks so
you.
much. Why don't we have one quick question from Deborah, and then we can of course follow up with these really important questions after the next talk. Why don't you take it away, Deborah?
Sure. Kate, that was actually wonderful. It's such a fascinating topic. I wondered within the WISE if you've had an opportunity to look at markers of immune activation in bacterial vaginosis.
Oh yeah, a great question. Some folks have been looking at that. I have only recently been kind of involved in that work. I think there was a recent publication we put out looking at glycogen and BV in the context of HIV as well. But I am blanking. We actually looked at cervicovaginal cytokines and chemokines here as well. None of them were really significant, so I haven't included any of them, unfortunately, which is interesting because there was another recent paper that was kind of actually doing a similar study, but in women without HIV. And they also found not so much of a difference with cytokines and chemokines. They were looking at L-cil and H-cil. But at the point of invasive cervical cancer, that's where you saw a lot of these cytokines and chemokines really come up. So it's interesting in terms of, well, we know there's some immune processes happening clearly. I mean, it's not, so maybe this is just an issue of like, we're not picking it up in the CBL or not looking for the right markers or something. But sorry, I digress. You were talking about, there's definitely inflammation. I am not, I think, what's his name? I'm totally blanking. Oh, it'll come to me in a second. There's other folks essentially leading that in the WISE.
Good.
But yeah, a lot of inflammation associated with BV. Hope I answered.
Thank you. Thank you.
Great. Well, thank you. And thanks again, Dr. Michelle. This was wonderful. And why don't we turn over to Dr. Ghosh, and then we'll come back to some other questions a little bit later. You should be able to share screens. Mimi? Oh, very good. Great. Thank you so much. And thanks for that lovely talk.
Thank you.
Okay, so am I okay? Well, thank you for inviting me to talk about this topic of interest today, which I like very much, HIV associated with inflammation. And since I work on the female reproductive tract, I will focus on that, although HIV is associated with many different kinds of inflammation. Let me turn the video off while I talk. It distracts me. Okay. So I thought I will start with what they call a word cloud, because it kind of, it's very interesting. So this is a paper from 2018 where they searched for the word HIV and corresponding associated factors. And then they plotted it out in this nice image where the size of the text is proportional to the number of publications on the topic. So I just wanted to start with that because inflammation is right in the middle as an associated factor for HIV. So it is very important. Other things you see in big letters are target cells, STIs, cell activation, which are all associated with inflammation. And then bacterial vaginosis, as Kate just talked about that. So women are disproportionately affected by HIV, and globally, there's still a lot of women who are infected and getting infected. And for women in the global scale, HIV is primarily a sexually transmitted infection. However, HIV does not actually efficiently infect the female reproductive tract, which is really interesting. So if the female genital mucosal barrier is intact, then there's like a 99.9% efficiency and nothing gets through. The problem happens when this barrier is breached, mostly by inflammation, immune activation, just physical trauma, some of those things I'll talk to you about today. So what do I mean by the HIV does not efficiently infect the tract? So if you look at this paper, it's a table from an old paper, 2008 actually, but what they did was they figured out transmission probability per exposure event from different routes of infection. Per exposure event means per act of sex. So female genital tract, the transmission efficiency is, or probability is 1 in 200 to 1 in 2000, which is pretty low all in all. If you consider blood infection, so from blood products or sharps, it can be as high as 95 out of 100. So the tract is efficient, the barrier is efficient in blocking infection when the barrier holds, basically. So let's look at the barrier a little bit. So here is a schematic of the lower tract, the vagina and the ectocervix, and the upper tract, which is the uterus and the fallopian tube and the endocervix. As you can see, the lower tract has multiple layers of epithelial cells. So it's kind of a thick layer of cells, which is difficult to breach. Whereas the upper tract is a single layer of columnar epithelial cells and much easier to breach. It is thought that HIV kind of enters through this zone of transition between the ecto and the endocervix. This is what actually a nice barrier looks like under the microscope in the lab, for those of you who don't look at that kind of images usually. So in this control picture here, you can see this nice green cobwebby cobblestone pattern. That is an intact barrier. So that looks beautiful. And then when you add HIV, you can see all this definition just disappears, and it disappears very quickly. So that is a disrupted barrier. And as you may imagine, as the barrier is disrupted over time, the virus translocates and enters the cell. So this is showing you 2 hours to 48 hours, and the virus keeps increasing. So how does inflammation relate to HIV? There are many mechanisms, and I'm going to give you a quick overview of 3 of those. So pre-existing inflammation due to STIs, barrier breach due to physical trauma, and these are kind of related. And then life cycle stage, which I'm going to talk about, immature cervix in adolescent girls. So I thought I'll put up one slide of mechanism of inflammation because I think about inflammation every day, but most of you guys probably don't. So I'll just give you a quick cartoon. It's not the best, but here is a schematic of the female reproductive tract, and this is like a tissue injury, let's say. So what happens is the cells here at the injured area, they start sending out signals, chemoattractants. So these are chemokines. They're inflammatory in nature. So these signals start here at the site of injury and kind of form a gradient to attract immune cells from elsewhere, from the bloodstream, to come rushing to the rescue, to clean up the pathogen, to heal the wound. So it's like a call for help. And where do these cells come from? These cells are coming from the bloodstream. So here are white blood cells kind of floating around in the blood. When they sense a signal like that, the capillaries kind of widen, and these cells, as you can see in this picture, the capillaries become leaky, and these cells squeeze out of the blood vessels and migrate to the site of injury by following this signal. As that happens, you have heat, redness, tenderness, swelling, pain, fever. These are all the clinical symptoms that you feel, which is basically this inflammatory process. This is what you feel from that inflammatory process. So this is how the mechanism works. Now, if you look at this picture of a pre-existing STI situation, so here is a barrier that's intact, except this breach here. So we are seeing a bunch of different STIs here, chlamydia here, BV causing bacteria, gonorrhea, HIV, HPV. When the barrier is intact, as I said, nothing is getting through. So they're kind of all hanging out there. And then the barrier is breached, and some of the HIV starts getting through and causes an inflammatory response, just as we talked about. So this results in a production of cytokines, chemokines. All these immune cells come to rescue, show up at the mucosa where the virus is hanging out. And then all these immune cells are actually target cells for HIV. So for HIV, that's what happens. So when the immune cells actually come to rescue, those are highly infectable by HIV, and therefore you have an event of HIV infection. So this is kind of the overall knowledge right now, that pro-inflammatory cytokines and chemokines attract target cells. They're just coming to help. But in the end, the HIV ends up infecting them, and so HIV replication is increased. So what we know is in HIV uninfected women, local genital inflammation is associated with high rates of HIV acquisition. And in HIV infected women, local inflammation correlates with genital shedding of virus irrespective of plasma viral load. So your plasma viral load can be undetectable, but you may be shedding in the genital tract. So the next related point is the breach. So I keep talking about, the inflammation happens because of the breach. How does the breach happen? The breach happens fairly frequently. So wounding and barrier breach is due to trauma, and trauma sounds like a terrible word, but actually in consensual and non-consensual sex. But just consensual regular sex can result in abrasions, tears in the cervical vaginal tissue, and this will end up stimulating inflammation. So the first point I want to tell you is about how sexual activity actually stimulates biomarkers in the reproductive tract. So sexual activity causes tear and abrasions in cervical vaginal surface. And then the virus or the pathogen is coming in with the semen, right? So the semen directly has a lot of roles to play in this. So semen alters pH in the vagina, semen stimulates cytokine and chemokine secretions by vaginal epithelial cells. That's how the sperm moves in. So the semen has to do that. Semen also increases an influx of immune cells. So these CD4 T cells and macrophages, they are target cells for HIV. So all of this can instigate, greater HIV replication. There were some papers earlier showing that inflammatory biomarkers are actually higher in adult women who are sexually active compared to those who are sexually abstinent. So just the immune microenvironment of those sexually active are very distinct. There was also this really interesting paper in 2016 where they collected baseline samples from women, and they had them come back to the clinic either 2 to 6 hours after sex or 10 to 14 hours after sex with their partners. with or without condoms. So very interesting. So what they find is when they have condomless sex, right after sex between 2 hours and 14 hours, the pH changes in the vaginal environment, and all these immune biomarkers also change in the vaginal environment. So just the sexual activity, not only does it create some tears and abrasions, the immune environment completely changes. So this is consensual sex with a partner. We have studies where we have sampled women who are survivors of sexual assault, and our controls were those with no sexual assault exposure are recent cases. So in these pictures, if you just pick any of them, let's look at IL-1 alpha here in this corner. So controls with no ever sexual assault exposure. Recent cases were those who had sexual assault exposure about in the last 3 months, and acute cases were those who had an exposure experience in the past 72 hours. So if you look at these graphs, all the acute cases, which are all the greens in all 4 of these graphs, had higher inflammatory biomarker compared to the recent cases and compared to the controls. Again, showing that right after sexual activity, there is a change in the immune environment, immune biomarkers in the female reproductive tract. So my third point to make is life cycle stage and how immature cervix is associated with HIV acquisition. So immature cervix is a normal phenomenon. It's called ectopy. It's found in adolescent girls. Now, adolescent girls are a very high risk group for HIV acquisition. So what do we know about that? Epidemiological studies have linked immature cervix or ectopy to increased HIV risk. Adolescent girls worldwide are also at high risk for coercive sex or early sexual debut, early child marriage, all of these things. But when they have sex early, they undergo greater genital tract injury than adult women. Immature cervix is easier to breach, and in addition, there's higher baseline inflammation in adolescent genital tract compared to that of adult women. So then this combination of genital tract injury and inflammation, presence of inflammation, can definitely enhance HIV, risk of HIV susceptibility. So let me show you some pictures about what I mean here. So this is the schematic I showed you before, where the lower tract up to the ectocervix has multiple layers of, multiple layers of squamous epithelial cells, and then the upper tract is the thinner single layer columnar epithelial cells. In adolescent girls, this ectocervix area is actually thinner single layer columnar epithelial cells rather than this thick layer. So if you look at the picture here, the top picture is 67% ectopy. This is an immature cervix. And the redness, so clinically you'll see it like this. So this redness here is actually, if you look at it at the cellular level, it's a thin layer of epithelium, as opposed to the adult mature ectopy, no ectopy cervix picture here, where this is going to look more like this under the microscope. So this is what breaches very easily because it's just one layer of cells, and therefore that is a direct risk of easier breach, and then you have inflammation underneath. So that links that mechanism, one of the biological mechanisms why adolescent girls may be more at risk for HIV and other STIs. From the same paper, they showed the baseline inflammation data. I thought that's really interesting. So the 2 red boxes are really, the first one here is comparing median levels of biomarkers from those with an immature epithelium, so ectopy. And the second red box here is showing you the data from mature epithelium. And there's a whole bunch of biomarkers. Most of them are inflammatory. So if you just look at any of them, basically the first box versus the second box, you'll see those with the immature epithelium had higher levels than those with the mature epithelium. So here is the data for higher baseline inflammation. We also found, in addition, that if we look at just within the adolescent population, depending on whether they have never been sexually active or if they are sexually active currently, there is a difference in immune biomarker profile in their genital tract. So we found that TNF alpha, which is an inflammatory cytokine, is higher in sexually inactive girls than in sexually active girls. And the sexually inactive have never been active sexually. We also did some heat map correlations, which we found interesting data here. So what we are doing here is basically we're lining up all our biomarkers on both sides of this triangle here. and percent ectopy is the last column, which I'll talk about in a second. So when we're lining up all the biomarkers here, basically we're just cross-comparing and looking for associations. So in these heat map patterns, red is positive correlation, blue is negative correlation, darker the color, stronger the association. And whenever there's an asterisk or more than one, that indicates strong significance or strength of significance. So this one is sexually active. This one is sexually inactive. If you just glance through, you can see that the correlation pattern is not the same. In addition, if you look at the ectopy column, percent ectopy, which is the last one for both of them, you can clearly see that the ectopy, extent of ectopy correlates very differently with the immune milieu, all the biomarkers that are there in the sexually inactive compared to the sexually active. Again, suggesting that just sexual activity does affect the biomarkers, and that does link to risk of HIV and other STIs. So last few slides, I'm going to talk about hormones, because everything we talked about so far are regulated by sex hormones, estrogen and progesterone primarily. So these are the master regulators of all things immune in the female genital tract. So top picture here shows you sort of adolescent reproductive age and menopause, and how the estrogen and progesterone, very roughly, how they kind of fluctuate. Because they fluctuate like this, HIV risk is very distinct throughout the life cycle of women. And I won't go through all of these here, but all of these functions change through adolescent reproductive age and menopause, with the common functions, shared risk factors being the things we are talking about: inflammation, epithelial barrier breach, and as Kate just talked about, the microbiome alterations. We even studied 4 women. We followed 4 women, so each color is 1 woman, through 2 to 3 cycles. The data here is from 2 cycles, so their own menstrual cycle. We just sampled them proliferatives of the first half of the cycle, mid-cycle, and the last half of the cycle, to see how biomarkers, inflammatory biomarkers, fluctuate during the menstrual cycle in the same woman across consecutive cycles. As you can see, some just don't fluctuate all that much, whereas others fluctuate quite a lot, and they don't even necessarily consistently fluctuate the same way through different cycles. This complicates how we do research on female genital tract, because everything is very flux, everything changes very easily. So any HIV risk we want to predict, for example, we have to take into account all these natural fluxes that are happening. This, I thought, is a really nice summary from 2018 review on sex hormones and HIV risk in women. So it's just showing the menstrual cycle in reproductive age women. The first half is follicular phase, second half is luteal phase. This is estradiol dominant, this is progesterone dominant. And it just kind of lists the immune functions that change during each of those phases. And generally, the data so far shows that HIV susceptibility is lower in the first half, or the follicular phase of the cycle, and higher in the second half, or the luteal phase of the cycle, which is dominated by progesterone. So this is my last slide. So I'm talking about inflammation and how that is bad for HIV acquisition transmission. So why do we not stop inflammation? And can we just control that, and then, life will be great? So let me start with this model again from an older paper, but inflammation is sort of the center here, so I like that. So inflammation is the center point here, and then the arrow here shows outcomes for HIV infection, which are increased replication, increased target cells, decreased barrier, increased microbial translocation, increased immune activation. So inflammation kind of translates into bad. And this is the part we talked about today on co-infections, barrier disruptions, and BV that Kate talked about. So there's both indirect and direct pathways of inflammation stimulating HIV infection outcomes that are bad for the host. So what if we block that? Then, this thing should be controlled or manageable. Unfortunately, this has not really worked out so far. So there has been studies trying to block 1 or more STIs. By block, I mean treat STIs in places where there are high rates of STIs, to see if that reduces the HIV acquisition rates in those populations. And it really hasn't. We haven't really seen exciting data so far, which is unfortunate. One of the reasons may be because STI treatment is always related to symptomatic infections because, obviously you see discharge or you see symptom and that's when you treat them. Whereas asymptomatic infection seems to be just as inflammatory in the sense they do all of these things. They stimulate the immune system just as symptomatic infections would. Either way, treating HSV, for example, or Trichomonas hasn't really, or BV, as Kate said, hasn't directly translated into reducing HIV acquisition in a given population so far. And this is kind of where the field is on inflammation and HIV. So I'll end there, and I have a lot of people to thank, particularly the names in red are those who participated in the studies that we talked about today. My wonderful collaborators in the department and CFAR, and these are collaborators elsewhere. Again, the studies I described today, these are collaborators for that. And this is my funding for the studies I talked about today. So thank you.
Excellent. Let's do a virtual round of applause for this outstanding talk. Thank you. This was wonderful and beautiful timing to you both. So thank you so much. Why don't we take a few questions for Mimi first about her talk, and then maybe 3 questions. It'll be perfectly balanced, and then we can open it up to questions that are cross-cutting. Does anyone have any question?
I have a question.
Yes.
Hi, Mimi, it's Larry.
Hi.
I'm just curious. I know that the American Academy of Pediatrics divides adolescence into 3 stages. So, what stage of adolescence, were you referring to in your research?
Yeah, so these are the older adolescent girls. So the sexual active, inactive. So we kind of want them to have menstrual cycle already, relatively regular menstrual cycle, or that's what this study was. So they're kind of I think stage 3, so closest to the adult population. We did have a couple of much younger girls there, but mostly this population has been 15 is the average age for this population.
Thank you.
That's a really good question.
Jean, did you have a question?
Yes, thanks Mimi for a great talk. My question is around change in pH and the mucus. Is there sort of a change in the mucus when you increase pH? And what role does thinning the mucus have in all of this?
Yeah, so the mucus actually cycles through the menstrual cycle. I don't have the picture. I do have the picture, but I didn't have it in the talk. So during the ovulatory period where you're, if you're going to get pregnant, that's when you're going to get pregnant. The mucus thins to almost nothing, so the sperm can get through. After that, the mucus starts thickening. So the body assumes you have had a successful conception. So the mucus actually thickens to protect that immune environment. The pH offhand, I cannot remember. Do you know, Kate, how the mucus relates to?
It does change with pH, although
It does. I can't remember which way. It does change.
I think with increasing pH, you have a thinning of the mucus as well. Although the mechanism exactly, it's not like directly lactic acid.
You know what else will do it? Because the normal acidity, it's normally the vaginal environment is acidic, so the pH is low. But obviously STIs enter with seminal fluid and sperm and all that. So when that happens, that fluid is highly alkaline. So it raises the pH immediately. And normally the vagina is protected by the low pH, but as soon as you have that sexual interaction, the seminal fluid is high pH, and that actually enables the sperm to go through because that's the reproductive biology side of it. So that also increases the pH immediately, and the mucus is thin at the time because you want the mucus to be thin to allow the sperm to go through. So thinking about some of this, it's like physiologically designed around reproduction. And then we are thinking of it from the perspective of STIs, but really physiologically you have to think in terms of reproduction.
Wonderful. Does anybody have another question for Mimi? No? Well, I'm gonna, I have a cross-cutting question. This is probably naive, but I'm an epidemiologist, so this is not my lane, so you'll have to forgive me. But I'm interested, so I'm thinking about the vaginal microbiome part and then the inflammation part. And it does seem like I'm interested in saying there seems to be an interesting problem that if you are trying to defeat the inflammation from STIs by giving antibiotics, that would also seem to have a concurrent effect on changing the vaginal microbiome, perhaps. So then is it possible that the reason you don't see an impact of the STI treatment in having the desired effect is because you're, I don't know, disrupting the vaginal microbiome because of antibiotics, or does it not really work that way?
You are probably taking out one level of protection that way, right? Because you're disrupting so antibiotics will take out the lactobacillus. That's why you would develop yeast infection if you take antibiotics for a long time. So you're definitely taking out that one layer of protection. There are other mechanisms, obviously. So those are not as affected by antibiotics, but that definitely doesn't help. Kate, you have other things to add to this, probably.
No, I mean, yeah, pretty much agree. It's just, it's just very fascinating, the kind of interaction and symbiosis. I kind of alluded to this in my talk too, but I feel like we're just at the beginning of understanding. I mean, some of these drug metabolism studies of the vaginal microbiome are just at its infancy. Some of the first studies looking at how diet affects the vaginal microbiome have just been recently published. So we're just really at the beginning of even understanding what is it breaking down, what is it producing. I mean, it's, we don't classically think of, sorry, I'm digressing a little bit, like fiber intake affecting your vaginal microbiome as well, but it's just like we're just at the infancy of it, and I look forward to learning more about as this field really progresses. Anyway.
In the early days, we were thinking more about presence of lactobacillus. I mean, we still are, but now the field is moving more into the function and focusing on what it breaks down and what it does and how it interacts with the other things. And that aspect is new, really. New.
It's fascinating how in one part of the body, there can be so many things going on. And if you change one, it's not an easy answer about how that will, impact risk. I think Alan had a question.
Yeah, thanks for the great talks. It's sort of like Manya's question. In listening to each other's talks, from your perspectives, what could be some of the interesting questions that could be posed in the other person's field?
We're kind of the same field, Alan.
It's funny. Yeah, I agree. But looking at your presentation, it got me thinking about the cytokine and chemokine levels throughout the cycle, and how it seemed like for IL-8, I mean, big player, right? But there seemed to be almost like it didn't change, but there was also like different levels for women across the cycle, and it made me think if there was it'd be really interesting to do a longitudinal study, and maybe someone has done this, but I don't think I've seen it, of women normally cycling and then kind of looking at maybe even using like a group-based trajectory model, or what's the other one, latent class or something like that, to kind of get like these overall patterns. Are there women that kind of fall into, because I'm thinking, what's that? The concept of like hysteresis, where if you're coming from a low to a high of IL-8, or well, IL-8 didn't change, but SDF1, low to high, high to low, even if you're at the same concentration at some later point, that coming from high to low might kind of impact things. And I'd be interested to see. I need to maybe do some searching on that also, but I don't know that I've seen anything like that, and that'd be really interesting.
Also, I think there's the gap of when we do lab studies, our numbers are small and we can see people are fluctuating wildly, but translating that into putting number of studies together to see, in 2,000 people, how many they're fluctuating wildly, but maybe that fluctuation is within a range, and that is a range. That's missing. That has not been done. And that just complicates understanding the field, because we have so many biomarkers. We're measuring like 20, there's 200. So
yeah, absolutely.
Thank you.
Well, I think we are just about the end. This was a fabulous set of presentations. Thank you both so much. We're very grateful. And thanks for the wonderful conversations. This was a great CPS conversation. We're happy that you were with us, and thanks to the audience participants for this great conversation with our wonderful speakers. I don't know, Sam or Larry or Alan, if you want to say any parting words.
Well, I want to say thank you very much to both of you. I'm a sociologist, so I do not have a clinical background in the way that both of you started your presentations. It was very understandable to me, so I appreciate that. Thank you very much.
Thank you. We were attempting to do that. I'm glad that, yeah, that came through.
Excellent.
Thank you.
Thank you, everyone. Thanks for
All
asking us to
right.
speak.
All right. Well, thank you so
Bye.
much and have a great day. We'll see you next month, same time and same station. Thanks very much, everybody.
you.
Bye.
Bye.